Prokaryote.
The prokaryotes are a group of organisms whose cells lack a membrane-bound nucleus. Those organisms whose cells have a well-defined membrane-bound nucleus and organelles are called eukaryotes. Most prokaryotes are unicellular organisms, although a few such as myxobacteria have multicellular stages in their life cycles or create large colonies like cyanobacteria. The word prokaryote comes from the Greek pro before, and karyon nut or kernel. Prokaryotes do not have a membrane-bound nucleus, mitochondria, or any other membrane-bound organelles. In other words, all their intracellular water-soluble components are located together in the same volume enclosed by a cell membrane, rather than in separate cellular compartments. The division between prokaryotes and eukaryotes reflects two distinct levels of cellular organization rather than biological classification of species. Prokaryotes include two major classification domains of life, the bacteria and the archaea. Archaea were recognized as a domain in 1990. These organisms were originally thought to live only in inhospitable conditions such as extremes of temperature, pH, and radiation but have since been found in all types of habitats.
Relationship to eukaryotes.
The division between prokaryotes and eukaryotes is usually considered the most important distinction or difference among organisms. The distinction is that eukaryotic cells have a true nucleus containing their DNA, whereas prokaryotic cells do not have a nucleus. One criticism of this classification points out that the word prokaryote is based on what these organisms are not. They are not eukaryotic, rather than what they are, either archaea or bacteria. Another difference is that ribosomes in prokaryotes are smaller than in eukaryotes. However, two organelles found in many eukaryotic cells, mitochondria and chloroplasts, contain ribosomes similar in size and makeup to those found in prokaryotes. This is one of many pieces of evidence that mitochondria and chloroplasts are themselves descended from free-living bacteria. This theory holds that early eukaryotic cells took in primitive prokaryotic cells by phagocytosis and adapted themselves to incorporate their structures, leading to the mitochondria we see today. The genome in a prokaryote is held within a DNA-protein complex in the cytosol called the nucleoid, which lacks a nuclear envelope. The complex contains a single, cyclic, double-stranded molecule of stable chromosomal DNA, in contrast to the multiple linear, compact, highly organized chromosomes found in eukaryotic cells. In addition, many important genes of prokaryotes are stored in separate circular DNA structures called plasmids. Prokaryotes lack mitochondria and chloroplasts. Instead, processes such as oxidative phosphorylation and photosynthesis take place across the prokaryotic cell membrane. However, prokaryotes do possess some internal structures, such as prokaryotic cytoskeletons, and the bacterial order Planktomycetes have a membrane around their nucleoid and contain other membrane-bound cellular structures. Both eukaryotes and prokaryotes contain large RNA protein structures called ribosomes, which produce protein. Prokaryotic cells are usually much smaller than eukaryotic cells. Therefore, prokaryotes have a larger surface area to volume ratio, giving them a higher metabolic rate, a higher growth rate, and as a consequence, a shorter generation time than eukaryotes. In 1977, Carl Oes proposed dividing prokaryotes into the bacteria and archaea, originally eubacteria and archaebacteria, because of the major differences in the structure and genetics between the two groups of organisms. This arrangement of eukaryota, also called eukarya, bacteria, and archaea is called the three-domain system, replacing the traditional two-empire system.
Sociality.
While prokaryotes are considered to be strictly unicellular, most are capable of forming stable aggregate communities. When such communities are encased in a stabilizing polymer matrix, slime, they may be called biofilms. Cells in biofilms often show distinct patterns of gene expression, phenotypic differentiation, in time and space. Also, as with multicellular eukaryotes, these changes in expression appear to often result from cell-to-cell signaling, a phenomenon known as quorum sensing. Biofilms may be highly heterogeneous and structurally complex and may attach to solid surfaces, or exist at liquid-air interfaces, or potentially even liquid-liquid interfaces. Bacterial biofilms are often made up of microcolonies, approximately dome-shaped masses of bacteria and matrix, separated by voids through which the medium, for example, water, may flow relatively uninhibited. The microcolonies may join together above the substratum to form a continuous layer, closing the network of channels separating microcolonies. This structural complexity, combined with observations that oxygen limitation, a ubiquitous challenge for anything growing in size beyond the scale of diffusion, is at least partially eased by movement of medium throughout the biofilm, has led some to speculate that this may constitute a circulatory system, and many researchers have started calling prokaryotic communities multicellular, for example. Differential cell expression, collective behavior, signaling, programmed cell death, and, in some cases, discrete biological dispersal events all seem to point in this direction. However, these colonies are seldom if ever founded by a single founder, in the way that animals and plants are founded by single cells, which presents a number of theoretical issues. Most explanations of cooperation and the evolution of multicellularity have focused on high relatedness between members of a group, or colony, or whole organism. If a copy of a gene is present in all members of a group, behaviors that promote cooperation between members may permit those members to have, on average, greater fitness than a similar group of selfish individuals. See inclusive fitness and Hamilton's rule. Should these instances of prokaryotic sociality prove to be the rule rather than the exception, it would have serious implications for the way we view prokaryotes in general and the way we deal with them in medicine. Bacterial biofilms may be 100 times more resistant to antibiotics than free-living unicells and may be nearly impossible to remove from surfaces once they have colonized them. Other aspects of bacterial cooperation, such as bacterial conjugation and quorum sensing-mediated pathogenicity, present additional challenges to researchers and medical professionals seeking to treat the associated diseases.
Reproduction.
Bacteria and archaea reproduce through asexual reproduction, usually by binary fission. Genetic exchange and recombination still occur, but this is a form of horizontal gene transfer and is not a replicative process, simply involving the transference of DNA between two cells, as in bacterial conjugation.
DNA transfer.
DNA transfer between prokaryotic cells occurs both in bacteria and archaea, although it has been mainly studied in bacteria. In bacteria, gene transfer occurs by three processes. These are, 1. Bacterial virus, bacteriophage, mediated transduction, 2. Plasmid-mediated conjugation, and 3. Natural transformation. Transduction of bacterial genes by bacteriophage appears to reflect an occasional error during intracellular assembly of virus particles, rather than an adaptation of the host bacteria. The transfer of bacterial DNA is under the control of the bacteriophage's genes rather than bacterial genes. Conjugation in the well-studied E. coli system is controlled by plasmid genes, and is an adaptation for distributing copies of a plasmid from one bacterial host to another. Infrequently during this process, a plasmid may integrate into the host bacterial chromosome, and subsequently transfer part of the host bacterial DNA to another bacterium. Plasmid-mediated transfer of host bacterial DNA, conjugation, also appears to be an accidental process rather than a bacterial adaptation. Natural bacterial transformation involves the transfer of DNA from one bacterium to another through the intervening medium. Unlike transduction and conjugation, transformation is clearly a bacterial adaptation for DNA transfer, because it depends on numerous bacterial gene products that specifically interact to perform this complex process. For a bacterium to bind, take up and recombine donor DNA into its own chromosome, it must first enter a special physiological state called compartments. About 40 genes are required in Bacillus subtilis for the development of compartments. The length of DNA transferred during B. subtilis transformation can be as much as a third to the whole chromosome. Transformation is a common mode of DNA transfer, and 67 prokaryotic species are thus far known to be naturally competent for transformation. The development of competence in nature is usually associated with stressful environmental conditions, and appears to be an adaptation for promoting repair of DNA damage in recipient cells. Among Archaea, Halobacterium volcania formed cytoplasmic bridges between cells that appear to be used for transfer of DNA from one cell to another. Another archaeon, Sulfolobus solfataricus, transfers DNA between cells by direct contact. Frols al. found that exposure of S. solfataricus to DNA-damaging agents induces cellular aggregation, and suggested that cellular aggregation may enhance DNA transfer among cells to provide increased repair of damaged DNA via homologous recombination.
Structure.
Recent research indicates that all prokaryotes actually do have cytoskeletons, albeit more primitive than those of eukaryotes. Besides homologs of actin and tubulin, MREB and FTSZ, the helically arranged building block of the flagellum, flagellin, is one of the most significant cytoskeletal proteins of bacteria, as it provides structural backgrounds of chemotaxis, the basic cell physiological response of bacteria. At least some prokaryotes also contain intracellular structures that can be seen as primitive organelles. Membranous organelles, aka intracellular membranes, are known in some groups of prokaryotes, such as vacuoles or membrane systems devoted to special metabolic properties, for example, photosynthesis or chemolytotrophy. In addition, some species also contain protein-enclosed microcompartments, which have distinct physiological roles, for example, carboxysomes or gas vacuoles. Most prokaryotes are between 1 and 10 but they can vary in size from 0.2 to 750 Bior Margarita nambensis.
Morphology of prokaryotic cells.
Prokaryotic cells have various shapes. The four basic shapes are Korchdzi, spherical, bacilli, rod-shaped, Spirituanet, spiral-shaped, Vibrio, comma-shaped.
Environment.
Prokaryotes live in nearly all environments on Earth. Some Archaean bacteria thrive in harsh conditions, such as high temperatures, thermophiles, or high salinity, holophiles. Organisms such as these are referred to as extremophiles. Many Archaea grow as plankton in the oceans. Symbiotic prokaryotes live in or on the bodies of other organisms, including humans.
Evolution of prokaryotes.
The current model of the evolution of the first living organisms is that these were some form of prokaryotes, which may have evolved out of protocells. In general, the eukaryotes are thought to have evolved later in the history of life. However, some authors have questioned this conclusion, arguing that the current set of prokaryotic species may have evolved from more complex eukaryotic ancestors through a process of simplification. Others have argued that the three domains of life arose simultaneously, from a set of varied cells that formed a single gene pool. This controversy was summarized in 2005. The oldest known fossilized prokaryotes were laid down approximately 3.5 billion years ago, only about 1 billion years after the formation of the Earth's crust. Eukaryotes only appear in the fossil record later, and may have formed from endosymbiosis of multiple prokaryote ancestors. The oldest known fossil eukaryotes are about 1.7 billion years old. However, some genetic evidence suggests eukaryotes appeared as early as 3 billion years ago. While Earth is the only place in the universe where life is known to exist, some have suggested that there is evidence on Mars of fossil or living prokaryotes. But this possibility remains the subject of considerable debate and skepticism. Prokaryotes have diversified greatly throughout their long existence. The metabolism of prokaryotes is far more varied than that of eukaryotes, leading to many highly distinct prokaryotic types. For example, in addition to using photosynthesis or organic compounds for energy, as eukaryotes do, prokaryotes may obtain energy from inorganic compounds such as hydrogen sulfide. This enables prokaryotes to thrive in harsh environments as cold as the snow surface of Antarctica, studied in cryobiology, or as hot as undersea hydrothermal vents and land-based hot springs.